stability of the animals under field conditions or under grazing conditions. Because if with the animals are stall fed, then we can easily conduct a trial. But when the animals are under grazing condition, it is very difficult to conduct a trial because we can't calculate the actual intake of the animal. So for that condition, we conduct with the indicator method. We calculate the digestibility with the indicator method. In this indicator method, we use one indicator. You all are well aware with the indicator. So indicator is an inert reference substance. It may be called as indicator or as a marker. So it is an inert substance which cannot be reactive with any of the our body biological system, and it is inert to it. Neither it gets any it is having a reactive nature nor it is get digested. So this is known as indicator. So what are the ideal specifications of the indicator? How can we select an indicator? What are its characteristics? So it may be total indigestible and unabsorbable. So that indicator should be totally indigestible in our body system, because if it gets digested, we can't calculate the actual quantity of the indicator in the feces. So it should be totally indigestible and unabsorbable. So none of the part of the indicator should be absorbed in our biological system. No pharmacological action on the digestive tract. It should be inert to the digestive tract. So there should be no any reactive action should take place in our body by the indicator. It should be totally inert, means non-reactive in our digestive tract. It must mix intimately with and remain uniformly distributed in the digestive. So it should be mixed properly in the feed, and it should be uniformly. It should not be so heavy that it can accumulate at any one place. So it should be so light that it can uniformly distributed in total excreta, so that when we do our random sampling, we can estimate a portion of that excreta. We can take the portion of that excreta and we can estimate the total excreted indicator in the digested material. So it should be passed through the tract at a uniform rate and should be voided entirely. So this should also one of the important characteristics of the indicator, that it should be passed through the tract in a uniform rate, not so fast and not very slow. It should be passed through at a uniform rate. Secondly, it should voided entirely, means it should excreted entirely, completely. ऐसा ना हो कि कुछ पार्ट उसका रिटेन हो गया तो हम उसको करेक्टली डाइजेस्टिबिलिटी को हम एस्टिमेट नहीं कर पाएंगे। तो वो एक बार में हमने जितना इंडिकेटर उसको खिलाया है वो पूरा का पूरा कंप्लीट वॉइड होना चाहिए, एक्सक्रीट होना चाहिए। इट कैन बी रेडिली बी डिटरमाइंड केमिकली। तो केमिकली उसको अपन एस्टिमेट भी कर पाए। वी शुड एस्टिमेट इट केमिकली और by any of the technique, preferably be natural constant of the feed under test. So ये एक preference, it is a preference that it should be a natural constant. It is not a compulsion, but it is a preference. If it is a natural constant, we shouldn't have to add it in the feed. It is naturally present in the feed, so we can estimate it. So these are the ideal specifications of the indicator. What are all indicators we can use? So indicators are also of two types, that is internal or natural indicator. So what are the internals or natural indicator? It is a component of feed, example lignin, silica or acid insoluble ash. So these are the three indicator, natural indicators. Lignin, it is naturally present in the any of the feed ingredients. Silica is also part of a feed, and acid insoluble ash, except minerals, except um, ash, part of ash, the which is dissolved in the acid, is the acid insoluble ash. So that part all is also acts as an in natural indicator. Secondly, external indicator. External indicator which we have to add externally in the feed. So that is chromic oxide and carmine red. So these two are the external indicators, and the three components are the internal indicators. So all these are having all the specifications which we have just read before for an ideal indicator. That's why we have categorized it in the category of indicator. So digestibility over nutrient is calculated by estimating the concentration of the indicator in feed and feces, and that of the nutrient in the feed and feces without the quantitative collection of total feces and measuring.